We're here at Scripps Institution of Oceanography in La Jolla, California, and we're visiting with Dr. Dimitri Dehane, and he is a marine biologist and involved with bioluminescent organisms and studying bioluminescence. So maybe you could tell us a little bit about your research and your work that you do here at Scripps.
Yes, so bioluminescence is the production of light by a living organism, and most of you might be familiar with what bioluminescence is. You probably have been playing with fireflies if you are from the Midwest, but I study those underwater. And so I have different applications for bioluminescence. I use bioluminescence to assess environmental quality. So some animals actually produce light, and the light production always follows a certain pattern. If the pattern changes, that means the animal doesn't feel good or is being stressed. So you can actually assess how the animal interacts with the surrounding environment and how it interacts with changes of that surrounding environment. The other aspect that I do is that I study the chemistry involved in the bioluminescence, because bioluminescence involves a chemical reaction that places a luciferin and a luciferase together, and when they react together, then it produces light in presence of oxygen and sometimes another cofactor. When you have these chemicals, you can actually use them for biomedical or biotechnological applications. So there is a very high demand of those compounds that produce light, and I'm going after the ones that are basically unknown. And so what I do usually is that I go out in the field and to take those animals that are there at night. I don't work in the deep sea. I just go very shallow water, but at night, and I collect the animals that produce light and then bring them to the lab, and then I assess their chemistry using different instruments.
Now I know with bioluminescence, it's a chemical reaction, and perhaps a good analogy with that is a glow stick. So can you talk about how a glow stick works and how that is like what you study for your research?
Yeah, exactly. So the glow stick is basically contains two chemicals, one in the plastic tube and the other one in the glass tube inside the glow stick. And so those two are basically called the luciferin and luciferase, so the substrate and the reagent, the enzyme. And so when they are mixed together, you will have production of light. And so when you actually break the glow stick, you will actually induce the mixing of the two reactives, and that will basically produce light. And so in that same case, you can actually use light to detect many different component or constituent or chemicals. So NASA, for example, uses chemicals from the firefly, which is not a marine organism but a terrestrial one, and the fireflies produce light using those two kind of chemicals, luciferin and luciferase, but they need ATP, which is the molecule which is essential for production of light in marine organisms. So ATP is basically the molecule that supports life, will be needed for that chemical reaction, and NASA uses that chemistry to actually test how soil collected from Mars will actually react. If the soil will produce light, then that means ATP is present in the soil, meaning that life was present at one point on Mars. So there is a diversity and a wide range of application of the chemistry of light production from marine organism or terrestrial organism. And so this is one of the fields that I'm trying to develop, trying to find maybe new applications and new chemicals that might actually be used in areas where there are none of those chemicals that can be used for different limitations.
Good. Now I know that you study certain marine organisms that are found here in offshore waters off of California. Tell us about which organisms you study and then how exactly you study them.
Yeah, so actually I collect, usually what I do is that I collect the organisms at night, and I will just describe you briefly what I do with them, and then I will bring you two examples of animals that I study.
Okay.
So usually I go at night and I collect the animals where they are out, because they usually go out at night when they can produce light and the light is visible, because otherwise during the daylight you won't see the light. So you collect them at night, which is involving diving and a lot of time in the field, which can be fun or can be exhausting. And then I bring back the animals here back in the lab, and then I sort them from whatever other animal they live with. And then usually what I do is that I study them, I look at them under the microscope, and I try to look at what part of the body produces light, and then I use different types of instruments to characterize actually the light production. First, I characterize the pattern of light production. Do they produce light with flashes, with a long glow? And I use a luminometer for that. And the luminometer basically is a box, a black box that will record all the light that the animal produces over time. It doesn't tell you what the color of the light is. So to know what the color of the light is, I actually use what we call a spectrograph. And a spectrograph is a kind of a spectrophotometer that will tell you which is the spectrum of the light. Is the light produced in the yellow, in the green, or in the blue? And this is important because the light production is actually made with the color that is the best propagated through the environment. So most of the terrestrial luminescent organisms will produce light in the yellow. The ones that are shallow water will produce light in the green, and the deeper ones will produce light in the blue. So it's always very interesting to see how these different type of organisms, depending on their habitat, will produce different color of light.
Right.
And so I use the spectrograph to do that. And once I have characterized the pattern of the light production, the intensities and the kinetics, and also the color using the spectrograph, then I will probably try to extract the chemicals. And to do that, I collect many of those animals together. I dissect them, I take them apart to see what part produces light, and then I usually grind the organism or part of those organisms into a vial to extract the chemicals. Of course, once you reach that stage, you have a soup of chemicals, and you have to basically separate them. And to separate them, I use what we call an FPLC, which is a chromatography system that basically will separate the different proteins that are in the grinding solution, and I will separate them and then sort them back according to which one can produce light. And so the FPLC system is extremely useful for my research. And I can then isolate the protein, or at least one of them, and then try to rebuild the chemistry in the lab. So in vitro, try to compare what the animal actually produces in vivo. So I'm trying to close the loop of mimicking what animals do, but trying to mimic it in little vials. And that can actually be very useful for different applications in biotechnology.
Very interesting work. Thank you very much for sharing that with us today.
You're welcome. Thank you.